Hello my dear aspirants, I hope you all are doing well. So today I am again back with a very important topic, which is your digestive system, and that topic is important for almost any kind of life science exams. It may be your NEET or it may be CSIR NET, Northeast Slet, or any kind of life science exam. It's a very important topic, so watch the video till end. And if you like my videos, then please don't forget to subscribe to my channel. And one more important thing I would like to share with you. If you want to take detailed coaching for CSIR NET or Life Science Slet, then you can comment down below. I used to provide detailed coaching, or you can contact in the given number. So watch the video till end. So as we all know, digestive system is a system of our body that is related with digestion of the food. That begins in the mouth itself, and then esophagus, stomach, small intestine, large intestine, rectum, and anus. And all entire this canal is known as alimentary canal, or you can call it GI tract. Apart from these organs, there are some accessory organs like the salivary glands, liver, gallbladder, pancreas. These organs are called accessory organs because they also assist in the digestive system. So if we come to the layers of GI tract, what are the different layers? So mainly there are layers like serosa, muscularis, submucosa, mucosa, and the inside one, that is the lumen. Coming in details, this is the serosa layer, which is the outermost layer. Serosa, it protects from outer wear and tear. So it provides protection as it is the outermost layer working like an wall. After serosa, the next layer is your muscularis, which is the layer of smooth muscles. It is made up of longitudinal and circular muscles. Remember, muscularis layer is made up of smooth muscles. Next layer is your submucosa, this one. Okay, submucosa, this layer is rich in nerve endings and blood vessels. As because it is rich in nerves endings, so it helps in receiving signals like the signals for contraction, relaxation, etc. because it is rich in nerve endings. The inner layer is the mucosa layer. Mucosa layer, it contains epithelium that has absorptive and secretory cell. So remember, the mucosa layer is associated with your absorption and secretion as it contains absorptive and secretory cells. The most inner layer, this one, is your lumen. Okay. Apart from that, mucosa also contain lamina propria and basal lamina, and the basal lamina here is known as the muscularis mucosa. Coming to the detailed parts of the digestive system, that begins with your mouth. Mouth, definitely it has teeth, it has salivary gland, it has the tongue. What is the role of the teeth? We all know teeth helps in mechanical grinding of the food. Okay. Then tongue. Coming to the tongue helps in mixing saliva with the food, okay, so that the food becomes easy to swallow, easy to digest. And also it secretes lingual lipase. Salivary gland, it secretes saliva, of course, and it consists of alpha amylase, which is also known as ptyalin. The role of alpha amylase enzyme is to convert starch into glucose maltose and maltotriose, because starch is the complex form, and if it is converted to the simpler form, then it will be very easy to digest. Mouth is overall, if we say, mouth is the region where carbohydrate digestion takes place. There is one particular term which is known as bolus. And what is bolus? Digested food mixed with the saliva. Okay, that is called bolus. Digested food is not like completely digested, but bolus can be called as the half digested and ready to be swallowed. The swallowing of bolus is called deglutition. The process by which we chew our food is known as mastication. So remember, these two terms are also important for your exam point of view. Next part is your esophagus. Helps in motility of the bolus. Now the bolus will have to enter. Okay, the bolus will now enter the esophagus and it will move to the next locations. So esophagus helps in motility because it helps in peristaltic movement. What is peristaltic movement? It is a rhythmic movement of the bolus, okay, of smooth muscles called the peristalsis. So peristalsis is a particular movement, okay, it is a particular rhythmic movement, and that movement is mediated by the esophagus. That movement helps the bolus to reach our stomach. if there is no peristaltic movement, then there would be a lot of difficulty in movement of your food from your mouth to the stomach. So a rhythmic movement, a smooth movement of the smooth muscle helps in movement of the bolus into your stomach. And so stomach is very important because it secretes lots of gastric juices. Okay, storage of food, of course stomach helps as a reservoir of food for a particular duration of time. Here, like in the mouth, digestion of carbohydrate take place in the mouth. So in the stomach, digestion of protein will take place. And there is another word called chyme. So what is chyme? Bolus plus the gastric juices. When we talk about gastric juices, that means secretion of stomach, then HCl comes to our mind, hydrochloric acid. So when the bolus, you know what is bolus? When the bolus will get mixed with the gastric juices, the juices of the stomach, then it will be now called chyme. Now the food is no longer called bolus, rather the food will be now called chyme. And remember, the chyme is acidic in nature. Next is our small intestine, which is also very important. So there are three different parts of the small intestine: duodenum, jejunum and ileum. So what is the main function of small intestine? So two different functions are the main one. Like small intestine is the main site of absorption. Ingestion of food has taken place. Now we ate our food, now our food reached the stomach. Now that is not enough. Food needs to be absorbed so that we get the benefit of eating, right? So the food needs to be absorbed. So the absorption will take place in the small intestine. Next point, small intestine also help in lipid digestion. Like carbohydrate digestion take place in the mouth. Similarly, lipid digestion will take place in the small intestine. And it is the main site of monomerization of complex molecules, because if they are not converted into monomers, then how will our body able to absorb them? Absorption is only possible if complex molecules are broken down into simpler molecules. Next is your liver and gallbladder, because liver will produce bile juice. Okay, bile juice, what is the role of bile juice? Bile juice can form micelle with the lipids or fat droplets and will convert large fat droplets into small fat droplets, increasing the surface area so that lipase enzyme can work upon it. Whenever we talk about lipids or fats, they are basically hydrophobic molecule. Okay, they are basically hydrophobic molecule. They are lipid loving, they are fat loving, but they are water hating. They will repel from water. So whenever in the aqueous environment, imagine our body is the aqueous environment. So at that time our bile will help in formation of micelle. Okay, the hydrophilic part will accumulate together and they will escape from the aqueous part of our body or the hydrophilic part of our body, and thus a micelle will be formed. Now what is the role of micelle formation? Micelle can convert large fat droplets. If imagine a very large fat droplet will be converted into small fat droplets, okay, with the help of bile juice. Surface area is inversely proportional to the size, so increasing surface area will help the lipase enzyme to degrade all those fat molecules. Next one is your pancreas. Pancreas also release many enzymes like proelastase, trypsinogens, nucleases, etc. Here important point is that trypsin enzyme, okay, trypsin enzyme, or if we can call it trypsinogen, it is the inactive form. Oh, remember, all the enzymes, these are not exactly enzymes, these are zymogens. Now what is zymogens? Zymogens are the inactive form of enzymes. Pancreas are releasing many zymogens which are not exactly enzymes, they are inactive form of enzymes. Trypsin is the one who will convert all these inactive enzymes into the active enzymes. But who will activate trypsin? Because see, trypsin was also in its inactive form, in its zymogen form, which is called the trypsinogen. Now trypsinogen is converted into trypsin with the help of an enzyme called enterokinase. So first, enterokinase is an enzyme produced by small intestine, secreted in the small intestine. At first, enterokinase will come, it will convert trypsinogen into the active form called trypsin, and then the trypsin will convert all these zymogens into its active forms. Like for example, the active form of proelastase is elastase. further digestion will take place. Till now we have studied a little bit about the digestion, absorption and all, but after that more digestion is required, and it will take place in the small intestine. As we all know, we have already studied that small intestine is the main site of breaking down complex molecules into monomers. So further digestion will take place in the small intestine with the help of enzyme. Maltase is an enzyme that will convert maltose into its monomer, that is glucose. Sucrase will be an enzyme that will convert sucrose into its monomeric simpler form glucose and fructose. Lactase is an enzyme that will convert lactose into its simpler forms, that is glucose and galactose. The next part is our absorption. So absorption, till now we have studied, we have eaten our food and the food has got digested in the stomach, and small intestine was also playing a role. Now after digestion, absorption is important, because unless and until the food is not absorbed into our cells, then nothing is going to happen. So absorption, it is also taking place in the small intestine. As we have already studied, small intestine is the site of digestion and absorption. So this part of the small intestine is called villi. There are different types of cells. These cell are called the epithelial cell, or you can call them enterocytes. Okay, next a type of cells are there which are called goblet cells, and these goblet cells will secrete mucus. Next type of cells are called enteroendocrine, and endocrine means related with your hormones. So these type of cells will secrete hormones, like secretin hormones will do regulatory functions. Another class of cells known as pennate cell, and these pennate cell have microcidal activities. With the help of enzymes lysozymes, this cell can help in eliminating the micro. If we talk about the carbohydrate absorption in the enterocytes, there are different proteins. These are nothing but they are proteins. Okay, these proteins are present in the membrane of the cell. These proteins are actually called transporters because they are transporting certain molecules. If we take example of SGLT21, this is a transporter. It is transporting sodium and glucose in the same direction. Transportation of sodium is also important because it is facilitating the transport of glucose. Then SGLT1 also helps in transportation of or absorption of galactose into the enterocytes. Again, absorption of sodium, that means transportation of sodium, is helping transportation of galactose into the cell. Another transporter known as GLUT5 is helping in the fructose absorption by doing facilitated diffusion. Here no other molecule needs to be transported because fructose is getting transported by diffusion, okay, along its concentration gradient without any use of energy. But facilitated means it is not directly entering into the cell, someone is helping. That someone is the GLUT5, and that's why it is called facilitated diffusion, not simple diffusion. If it was simple diffusion, then fructose would easily enter the cell without the use of GLUT5. Okay, next one is a GLUT2. This is helping in exit of glucose. GLUT2 is also helping in exit of galactose. GLUT2 is also helping in exit of fructose. Now inside the enterocyte, sodium is getting very high because with each glucose, with each galactose, sodium also has to enter, because entering of sodium is assisting the enter of glucose and galactose. Now the sodium is getting very high inside the enterocytes, which is not normal. So a pump known as sodium potassium pump, this pump is also, it is a protein, it will help in exit of sodium and entry of potassium. Okay, so this is the last part where we will study absorption of fatty acid and absorption of vitamin in the enterocytes. How absorption of fatty acid will take place? If it is a short chain fatty acid, then simple diffusion without the need of any protein, without the need of any energy, simply it will enter the cell. If it is a long chain fatty acid, then it has to form micelle with the bile salt and enter the enterocyte. Okay, if it is long chain fatty acid, then definitely it has to form a micelle. Okay, its hydrophobic part will come accumulate together, and then it has to enter the cells. In the enterocyte, fatty acid will reassemble into triglycerides in the smooth ER. Okay, and triglycerides plus protein will be called kilomicrons. Last one is your absorption of vitamin B12. Here we are only taking example of vitamin B12 when we are discussing absorption of vitamin. Remember, vitamin B12 is a vitamin that is not that cannot be produced by us. We get vitamin B12 with the help of microorganisms, only from microorganisms. First, our salivary gland will produce heptocorrin. Okay, heptocorrin, actually it will combine with vitamin B12. And what is the role of heptocorrin? It will protect the vitamin B12 from the HCl in the stomach, from the acid of the stomach. Our vitamin B12 will be protected if it is combined with heptocorrin. Then both of them will reach the small intestine. In the small intestine, the heptocorrin will be cleaved. Okay, now heptocorrin will go away. How it will be cleaved? By the proteolytic enzymes. Now vitamin B12 will no longer bind with heptocorrin. Vitamin B12 will bind with some intrinsic factor which will be released from the parietal cells. Vitamin B12 plus the intrinsic factor will bind to a receptor known as cubilin receptor. This is a receptor which will receive vitamin B12 plus intrinsic factor, and this is a receptor present in the enterocyte. Then receptor mediated endocytosis will take place. That means the receptor, okay, will the vitamin B12 will be endocytosed, means it will be taken inside the cell, and like that absorption will take place. So I hope you have you got the basic idea about the digestion and the absorption. This I hope can clear some of your basic doubts. I hope the class was helpful enough. If you like my videos, then please subscribe to my channel. Bye and all the best.